Hello friends, welcome to our channel. In this video we are going to see about atomic absorption spectroscopy. Atomic absorption spectroscopy is a very common technique detecting metals and metalloids in samples. It is very reliable and simple to use. It also measures the concentration of metals in the sample. Atomic absorption spectroscopy is an analytical technique that measures the concentration of an element by measuring the amount of light that is absorbed at a characteristic wavelength when it is passes through cloud of atoms. As the number of atoms in the light path increases, the amount of light absorbed increases. The history of atomic absorption spectroscopy is the first atomic absorption spectrometer was built by CSI, that is Commonwealth Scientific and Industrial Research Organization. The atomic absorption spectroscopy was first discovered by scientist Alan Walsh in 1954. Now we are going to see the principle of atomic absorption spectroscopy. The technique uses basically the principle that free atoms generated in an atomizer can absorb radiation at specific frequency. Atomic absorption spectroscopy quantifies the absorption of ground state atoms in the gaseous state. The atoms absorb UV or visible light and make transition to higher electronic energy level. The analyte concentration is determined from the amount of absorption. Concentration measurements are usually determined from a working curve after the instrumentation with standards of known concentration. In atomic absorption spectroscopy, we will see radiation source, nebulization of liquid samples, atomizer, monochromators, and detectors. It is a diagram of atomic absorption spectroscopy. In this diagram, halo cathode lamp, flame, nebulizer, test solution, monochromator, detector and data processor. First we will see radiation source. Halo cathode lamp are the most common radiation source in atomic absorption spectroscopy. It contains a tungsten anode and a halo cylindrical cathode. These are sealed in a glass tube filled with an inert gas. Each element has its own unique lamp which must be used for analysis. Then nebulizer. Nebulizers suck up liquid samples at controlled rate. Create a fine aerosol spray for introduction into the flame. Mix the aerosol and fuel and oxidant thoroughly for introduction into the flame. Then atomizer. Elements to be analyzed need to be in atomic state. Atomization is separation particles into individual molecules and breaking molecules into atoms. This is done by exposing the analyte to high temperature in a flame and graphite furnace. The atomizers most commonly used nowadays are spectroscopic flames and electrothermal. Then flame atomizers. Nebulizers suck up liquid sample at controlled rate and create aerosol spray for introduction into flame. To create flame, we need to mix an oxidant gas and a fuel gas. In most of the cases, air acetylene flame or nitrous oxide acetylene flame is used. Liquids or dissolved samples are typically used with a flame atomizer. Electrothermal graphite tube atomizer. It is used a graphite coated furnace to vaporize the sample. Samples are deposited in a small graphite coated tube which then heated to vaporize and atomize the analyte. The graphite tubes are heated using a high current power supply. Steps in electrothermal atomization: drying, pyrolysis, atomization and cleaning. Then monochromator. This is very important part in an atomic absorption. It is used to separate out all of the thousands of lines. A monochromator is used to select the specific wavelength of light which is absorbed by the sample and to exclude other wavelengths. The selection of the specific light allows the determination of the selected element in the presence of others. Then detectors. The light selected by the monochromator is directed onto a detector that is typically a photomultiplier tube whose function is to convert the light signals into electrical signals proportional to the light intensity. The processing of electrical signal is fulfilled by a signal amplifier. The signal could be displayed for readout or further fed into a data station for printout by the requested format. If you like this video, please like, share and comment. Thank you.